Hello everyone, thank you for joining me today. We're going to take a deep dive into one of the most critical components of pharmaceutical product development and regulatory compliance, that is ICH Q1B, photostability testing of new drug substances and products. This guideline helps us determine how light exposure impacts drug integrity, safety, and efficacy. Let's begin. Introduction to ICH Q1B. The ICH Q1B Guideline is part of the broader ICH stability testing framework and complements the ICH Q1A parent guideline. It provides a standardized international approach for evaluating how new drug substances and products respond to light. The key takeaway here is that light exposure can degrade pharmaceutical compounds, affecting both the quality and safety of medications. This guideline ensures a harmonized strategy across regulatory regions, the US, EU, and Japan, and plays a pivotal role in regulatory submissions, product labeling, and packaging design. Objectives of photostability testing. There are three major objectives of photostability testing. First, to identify if a drug substance or drug product is photolable, meaning it degrades upon exposure to light. Second, to ensure that light exposure does not compromise product quality, affecting its potency, purity, or safety. And third, to determine whether we need protective packaging, like amber bottles, aluminum blister packs, or specific label warnings. Essentially, we want to protect the medicine from sunlight, hospital lighting, or even shelf lighting, and ensure that it remains safe until its expiration date. Systematic testing approach. Photostability testing is approached systematically, and it typically follows this structure. Drug substance testing, first through forced degradation under exaggerated conditions, then through confirmatory studies under standardized light exposure. Drug product testing. This begins with exposing the drug product directly to light. If needed, we then test it in its immediate pack. And finally in the marketing pack, that's the full commercial packaging. This stepwise escalation helps us determine how well the packaging protects the product from light, and whether any changes are needed in formulation or packaging. Light sources for testing. The guideline allows two options for light sources. Option 1 uses a lamp that simulates natural daylight, like a D65 artificial daylight fluorescent lamp, or a xenon, metal halide lamp. Option 2 combines two different light sources, a cool white fluorescent lamp, and a near UV fluorescent lamp that emits light between 320 to 400 nanometers, mimicking sunlight's UV range. It's also important to filter out wavelengths below 320 nanometers, as those may cause unrealistic degradation not reflective of actual storage or usage condition. Photostability testing procedure. Now, let's walk through the actual test procedure. For confirmatory studies, the sample is exposed to a minimum of 1.2 million lux hours of illumination and 200 watt-hours per square meter of near UV energy. This mimics real-world light exposure over time. To ensure accuracy, we often place the sample beside a chemical actinometer like quinine, or use radiometers or lux meters to monitor the light intensity and duration. We also use dark controls, like foil-wrapped samples, to differentiate between light-induced and heat-induced degradation. Decision flowchart. This decision flowchart guides us through testing progression. If there is a formulation change, we test the product directly. If unacceptable changes are observed, we proceed to test in the immediate pack, and finally, the marketing pack. If light exposure still leads to degradation, we may need to redesign the formulation or choose better protective packaging. This logical flow ensures that testing is progressive and efficient, without over-testing unnecessarily. Drug substance testing. Let's now focus on the drug substance. We start with forced degradation studies to assess its overall photosensitivity. This also helps in the development and validation of analytical methods. If the compound is photostable, we can stop early. But if it's photolable, we proceed with confirmatory studies under standard conditions. It's important to use chemically inert containers. Ensure uniform thickness, not more than 3 mm for solids, and eliminate effects like sublimation or evaporation. Drug product testing. For the drug product, testing is typically sequential. Start by exposing the product directly. If degradation is observed, test it in the immediate pack, and then the marketing pack. For products in light impermeable packaging like aluminum tubes, direct exposure testing alone may be sufficient. Testing should also reflect real-world usage, especially for products like creams, infusions, or suspension. Sample presentation and analysis. Samples should be presented to maximize uniform exposure. Avoid interference from physical changes like melting or sublimation. After exposure, we assess physical changes like discoloration or turbidity. Chemical changes through assay and degradation profiling. Use validated analytical methods that are sensitive to photolytic degradation products. For solids, use composite samples, for example, 20 tablets or capsules, to ensure representative testing. Judgment of results. In forced degradation, the goal is to develop and validate methods, not set limits. In confirmatory studies, we assess the need for protective packaging, light-resistant formulations, labeling statements. Judgment must be based on data from formal stability studies to ensure the product remains within specifications throughout its shelf life. Quinine actinometry. The guideline also provides a validated procedure, using quinine hydrochloride as a chemical actinometer. This helps us confirm if the sample has received adequate UV exposure during testing. We measure the change in absorbance at 400 nanometers. For ampoules, a change of at least 0.9. For quart cells, at least 0.5. This gives us a quantitative confirmation of light exposure, supporting the reliability of our test results. Glossary of terms. To quickly recap some definitions: immediate pack, packaging in direct contact with the product; marketing pack, complete commercial packaging; forced degradation, harsh light exposure for method development; confirmatory studies, standardized light testing for regulatory purposes. Conclusion. To wrap up, ICH Q1B ensures that new drug substances and products are evaluated for photostability using scientifically sound and harmonized methods. It helps manufacturers ensure that their products are safe, effective, and compliant with global regulations. And most importantly, it safeguards patients from degraded or ineffective medicines. Thank you for your attention. I hope this gave you a clear and practical understanding of ICH Q1B photostability testing. I now welcome any questions, feedback, or discussions in comment box you might have.